Bioresource for discovery research in fisheries and marine science industries. First of all, let's say thanks to Allah Subhanahu wa Ta'ala who has given us guidance, happiness, health, and mercy, so we can attend and participate in this special event without any obstacle. On this special afternoon, we have several agendas as follows. The first agenda is opening, and then opening remark by the Dean of Fisheries and Marine Science Faculty, Jenderal Sudirman University, Dr. Isdi Sustadi A. And the next agenda is photo session. And the fourth agenda is presentation from speaker, Dr. Sanya Mihajlovic from Fraunhofer Institute for Molecular Biology and Applied Ecology, IMA, Branch for Bioresources, Gießen, Germany. And the next agenda, discussion, and the last is closing. Well, ladies and gentlemen, let's begin this event by reciting Basmalah. Bismillahirrahmanirrahim. And we move on the next agenda is opening remark by the Dean of Fisheries and Marine Science Faculty, Jenderal Sudirman University, Dr. Isdi Sustadi A. For Dr. Isdi, time is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Alhamdulillahirabbil alamin. Let's thanks to God, to Allah, that give us many advantage, and we are still in healthy, healthy, and we are able to resemble in this room. Our very distinguished lecturer, Dr. Sanya Mihajlovic, I'm sorry, from Gießen University, Germany. We are very proud and very happy that you can join us, and we hope that you can come to our university in a good and next time. Thank you very much for your willingness to be a lecturer today. And I think in Germany is still in what time? In 9 o'clock.
Exactly. Yeah. Very early.
Yeah. Okay. Thank you very much for that. And all of participants, all of lecturers in our faculty, and we have a very good opportunity today to have a lecturer from the highly expert of bioresources from Germany, and I hope that all participants can obtain this can obtain the lecture from her, and I hope that this is very beneficial to us. Yeah, beneficial to us, and most of all is we are waiting for another collaboration with Gießen University, and very close very close collaboration with Gießen University or in the future, in the very close future, maybe. Yeah. Okay. Because we have that. We hope that our pandemic will be over. Yeah, in our country. I know that in Europe it is over. Yeah, it is over, the pandemic, but yeah, because you have not take a mask anymore. Yeah. Okay. But again, thank you very much for our lectures from Marine Science Study Program and Marine Science Department to be for your very hard work and very nice work. And this meeting room will be is possible. Yeah. And again, thank you, Dr. Sonia. And then I know Dr. Sonia, I state officially and formally this public lecture begins. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Dr. Isti, for the speech. Okay, the next agenda is photo session. Please, all audience can open your video. And we can start to take a picture. We have
Please, everyone, maybe can open the camera. Then we have a really nice picture today.
we have 4 slides. Okay, we start for the first slide. You ready? 1, 2, and 3. Capture. And the second slide. 1, 2, and 3. Smile. And the third slide. 1, 2, and 3. And for the last slide. 1, 2, and 3. Okay, thank you for all audience. Okay, well, ladies and gentlemen, we arrived. The main agenda is presentation from the speaker. Before that, I will also introduce the moderator in this public lecture. Her name is Dr. Rernat Priyanti Biotech, and she is lecturer of Marine Science Department, Fisheries and Marine Science Faculty, Jenderal Sudirman University. So let's recall Dr. Rernat Priyanti Biotech. Dr. Rianti, how are you, Miss?
Hello, fine, thank you.
Okay, thank you for being a moderator for today's public lecture, Miss. And time is yours, Miss.
Okay, thank you very much, Ms. Riviani, for the nice introduction. I will say hello first again to Sanya. Good morning, Sanya.
Good morning.
Okay, today I'm really very happy because I can meet Sanya again. Okay, welcome to this public lecture. This public lecture held by Department of Marine Science, Faculty of Fisheries and Marine Science, Jenderal Sudirman University. We are in Purwokerto, Indonesia. Hopefully, today we will have like outstanding presentation from our lecturer, whom I believe she will enrich our insight and knowledge regarding to the topic this lecture. Okay, maybe short information to Sanya. Maybe usually we call Sanya just Sanya because maybe I will let you know that usually in Indonesia we call someone with something like in Germany her for example Frau something someone, but here in Indonesia it's not common if we just call Sanya, but I will let all the audience that is fine. Is it fine, Sanya, if we call you just Sanya?
Of
Okay,
course.
maybe it's more familiar. Then
Yes.
for the next, I will call you Sanya, and the other also can call Sanya. So actually, we have around 124 registered participants that mostly they come from our home university, Jenderal Sudirman University, but some of them also come from other university, also come from National Research and Innovation Agency or BRIN. Mostly here are students and also some of them also lecturer, researchers. Then I believe that they want to hear the really nice outstanding presentation today. Okay, before start the presentation, allow me to introduce our lecturer today. Okay, Sanya has research interest in classical and molecular microbiology, strain and data management, isolation, cultivation, detection, and molecular characterization of microorganisms from different ecogenesis, discovery of new strains and molecules relevant to bioindustry. She finished her diploma in molecular biology and physiology, and 2 times doctoral student in Spain and Austria. Then in 2010, she was the head of laboratory for microbiology in Serbia. Then continue as a postdoc in Institute of Molecular Genetics and Genetic Engineering in Serbia. Then 2017, she moved to Germany directly to Gießen as a team leader at Sanofi Fraunhofer's Natural Products Center of Excellence. And from 2020, she is a group leader of Biobank Fraunhofer Institute for Molecular Biology and Applied Ecology, Gießen, Germany. Okay. And today she will have like short presentation around 20 or 30 minutes about the strain collection as a bioresource for discovery research in fisheries and marine science industries. Okay, just short information from the all audience that later if anyone have any question, comments or suggestion to Sanya, so you can raise your hand or kindly write your question, comment or suggestion in the chat box. Okay, Sanya, I think we can start today. Sanya, the screen is yours.
Okay. Salam alaikum. Good afternoon, ladies and gentlemen.
Salam.
Good afternoon.
It's a real pleasure to be here today. I will start sharing my screen. I hope it's in a... Do you see my full screen? So am I in a right? Oh, it's
Yes.
completely wrong.
Okay.
A second. So you see my not
Yes.
full screen now.
Not full screen at the moment.
Now it's full, yeah?
Okay, yes. Okay.
Now it's okay?
Yeah.
Okay, good. So thank you very much. It's really a pleasure to be here today. As Rianti already introduced me, I will just skip this part of talk and give you some overview a little bit about the institute I'm coming from. And then I will be speaking about strain collection, so about the microorganisms, how you have to build collection, how you can utilize collection and so on, because I'm the person who is creating a collection. So besides being a molecular microbiologist, I'm also taking care, I like to say that's my baby. The second part of my lecture will be connected to some regulations, law stories. It's not so highly interesting to listen, but it is important, especially when it comes to your country, because this is one of the most biodiverse country of the world. So I think that we should speak a bit about that. So let me start. Fraunhofer Institute is applied research institution in Germany. So we have over 72 institutes over the whole country. And we are located, our institute is located on 4 spots in Germany. It is an institute of molecular biology and applied ecology. And we are located in Gießen. So we are a branch of Bioresources. What do we do in Gießen? So our mission is to translate the biodiversity of our bioresources into naturally involved products and services for the bioeconomy. So we work either with the whole organisms, microbes, bacteria, fungi. So we do, if you will, the biologics, or we work just with the part of organisms using nucleotides or proteins or their metabolites, meaning working on a RNA level, enzymes or natural products. And we are active in a different area of different industries. I like to call them bio industries or biotechnology. So green meaning agro field, agro sector. Red meaning more direction like veterinary, human medicine, and white biotechnology could be like related to fisheries, food and feed industries. And we have activities in all directions. Today, I will be speaking about our large strain collection. It is really a huge microbial collection that possesses over 110, now even over 120,000 microorganisms. Mainly, we are having bacteria, around 100,000, and the rest is fungi. And here on a picture, actually, you see our facility in Gießen. All these samples, actually, we had in past some collaboration with the industry, pharmaceutical industry, and they kind of outsourced their activity to us, and we become an owner of their collection. This collection is as you can see on this map, world map, it's almost completely blue. It means that at least one of the samples was isolated for some of the species in so many countries. I would say we have in total 131 countries that our samples are coming from. Very, very diverse. And this job actually, our oldest microbial sample dates from before First World War, meaning 1940. So very, very old. And the samples were collected from natural habitats over 7 decades, including soil, oceans, in extreme environments, plant, lichens, and so on. So any exotic place you can imagine on a world, we were there, not myself personally, but in past people collecting the samples. This is, I will not go into the detail here, but this is a composition of our collection. Mainly samples were isolated from soil, and that means that mainly we have in collection, when it comes to bacteria, we have actinobacteria the most, and the others are less present in our collection. From fungi, huge proportion, so almost 3/4 of our collection is still unknown because we didn't identify them by the species or genus level. Among our collection, there is also a small collection that is related to marine microorganisms. This collection counts over 1,000 samples, and mainly they were isolated from algae or different invertebrates, coral reefs, marine sediment, and so on. And mainly, as you can see on this graph, I'm not sure how I can make my presenter. Remote control. Just a second. No, it's too complex for me.
Usually you can find in the left side of yours, and there is a choice for laser pointer.
I don't see it. Doesn't matter.
Okay,
I'll
yeah.
just go ahead. So on this graph, you can see that mostly we have collected from these samples actinomycetes, fungi, and streptomycetes. So it's not like by taxonomy systematic, it's not like correct official taxonomy, but we call that in a praxis, actinomycetes and streptomycetes. For instance, these samples were isolated from this type of organisms. And these are just like an examples of plates, of like isolation, how it looked like. What we do now is in last 7 years, we had our focus a little bit moved in direction of insects. And we now start isolating gut microbiomes of different insects. Yeah, we are searching now for new underexplored species. And as you can see, this is like a work of 7 years, but we only have isolated around 2,000 microorganisms, with the hope to observe a huge proportion of, for instance, this acidobacteria or planktomycetes. But as you can see, it's really like a small percentage, which tells you that it's really hard to get like uncultivable stuff culturable. So I would be happy if especially students, and I know that on this lecture are so many students present, I would be happy if you get some basic understanding. What does it mean to possess strain collection? It's a long-term process, and it requires several steps to build a strain collection. It means that you first have to do microbial isolation. You can use different type of methodologies, culture dependent, culture independent, and this is the most critical step, long-lasting step, because you really have to be a good microbiologist to have a good knowledge to know how to do that. The next step is to characterize, because you want to know what you have isolated, and then you use different techniques, different methods, morphological, biochemical, and in parallel genetics. So you use different approaches. Once when you isolate, characterize your microbes, you need to preserve them. And there are different ways of preservation, either refrigerator using or deep freezing, freeze drying. Well, once when you created your collection, your aim is not to just keep these microorganisms. Your aim is to explore them, to utilize and to search for the products, to search for their function, how to say, that can be used in different industries, especially that human can benefit. Let's go a little bit. I will not go so many into the details, but still I will be sharing some details with you. What does it mean when it comes to this first step, microorganism isolation? So you take your sample. Rianti, I felt free to take some even photos from your doctoral dissertation, if you can recognize them. So you take some samples, you homogenize your sample, and then you start isolation. Yeah, it sounds very simple when I say you just start. It requires so many knowledge about, you have to know which type of nutrients you need in order to get your microorganisms grown. Yeah, well-known stuff you can find in the literature, different publications. But if you're really interested in something that is not very well known or not cultured so far, then it's a real challenge. And at the end, your aim is to come from your sample source, bioresource, to the pure cultures. Once when you received pure cultures, you go with you start to characterize your microorganisms. One of the first ways of characterization is using microscopy, so determining morphology. Either you use very simple light microscope, or you are very fancy and you use transmission scanning, electron microscopy, and so on, depending on your university or your company resources. Once when you determine that morphologically, you also want to know what type of characteristics biochemically your samples your microorganisms are having. So you apply different type of assays. And it all helps you actually to determine the species, to give I like to say very often, the next step is to give the name and surname to your microorganism. So you need to have like a full passport of, each microorganism has its own passport. That means that you need to characterize that most of the time simply by using 16S ribosomal RNA gene for sequencing in order to determine bacterial taxonomy, or 18S to determine fungi taxonomy. And very often, if you want to be very precise, you will be more focusing on ITS region, internal transcribed spacer, so highly variable sequence. And it all will help you actually to give a name and surname to your microorganism. That again will help you because you are a researcher, you have a specific aim, you want specific microorganisms in order to produce some compounds of your interest. What is also important to know, when you have, for instance, you collected 100 microorganisms, and you want to perform experiments and you want to find out about their biochemical characteristics, to find out which type of molecules these microorganisms produce. But it's a little bit hard if you have at once to work with, let's say, 100 microorganisms. Before you do that, you should actually look and check how similar, how identical, how diverse are these 100 microorganisms. To find out that, you will look at their genetic profile. That means that you can actually exclude redundancy. You can remove from your further work the organisms that are identical, because you don't need, in a science or in academia or the precise work, you have to have a replicate. That's completely fine. But you don't want to have like one organism repeating 20, 30 times. So to avoid all this, you apply so-called DNA fingerprinting that will enable you to differentiate your microorganisms or groups of microorganisms. If you're interested in more detail how to do that, you can read Rianti's publications, so you can find more details there. I didn't put them on a slide here, but she can share that with you later. And this is, you use different type of primers to amplify these repetitive elements of DNA. And at the end, you receive the genetic profile and you look at that profile and you find out how similar or how different your microbes are. You can do that in a very simple way, just doing a PCR, then running the normal gel electrophoresis, or you can be very fancy and use some capillary electrophoresis as we do. This is actually a photo of the things that we do in our laboratory. And but the end result is the same. Well done. So you have isolated, you have characterized your microorganism. That means that your next step is to do preservation. In our case, so here you are seeing the pictures of our biobank facility in Gießen. And we have over 500,000. Now we are even, the slides are very old. So we have now almost close to 1 million vials, physical, these cryo vials in our facility. Around 20,000 are freeze-dried or refrigerated, not all, but refrigerated, sorry. And we have 100 that are cryopreserved either at -80 or 100 for liquid nitrogen, so very low temperatures. Once when you are completely, at the end of this characterization and preservation, actually your research can start. Now you can start to use your microorganisms in discovery research. I named my presentation as the use of these microorganisms in fishery and marine science industries, but you can apply all microorganisms to actually different industries. They are an unlimited source for use to any industry. They are producing natural products that can be used really at the same time for different sectors, for fisheries, for pharma, for veterinary medicine, for plant protection, whatever you want. It depends on your purpose. It depends on what you are looking at. And how we do that at our institute, we have very well developed natural product discovery pipeline. Unfortunately, I missed to actually cite our publications, but in, I think, last 2-3 years, we have around 20 publications that came from work of our last 5 years, I would say. And all of them came actually from this pipeline. What do? We use our bioresource, as you can see on the top right of this slide. We use our biobank, our microorganisms, and we cultivate these microorganisms. So you have to have a good knowledge of the way how to cultivate them, because you want them to produce specific compounds. You're looking for the silent genes, cryptic regions. And you then extract the compounds they are producing. Then your next step is to do screening. You do different biological assays, screen, you're looking for activities. Once when you determine activities, you enter now, how to say, analytic pipeline. Now you want to know specifically to determine which compound actually gave you activity. So you're going from a strain to really active compound. And once when you determine the peak, the thing that gave you activity, you try to scale up to isolate that compound and later on to do structure elucidation. And once when you are finished with this package, you can actually try to, if you are, let's say, working at universities, then you can start contacting different companies, because you are at the stage that you have some so-called lead structures, and you can enter now negotiation with different companies and put your product on a market. I mean, it's still a long process to come there, but at least I would be happy if you get some impression how this process is actually complicated. Well, with this, I will close first part of my lecture, meaning how to build a strain collection. So the collection I showed you is a real connection from Gießen, the work we do here. And now I will focus on something that I think that is very important for any human being on this planet. I will be speaking now about implementation of Nagoya Protocol in strain collection or in general. And I will be probably repeating myself from time to time with the aim that you, after this lecture, keep in mind at least something what I said. Coming back a little bit, historical overview. Coming back to 1992, there was an Earth Summit where 3 so-called Rio conventions happened. Convention on biodiversity, convention on climate change, and desertification. So all of them had a goal to contribute to the sustainable development of Agenda 21. Agenda 21 means, I think it was kind of thinking of agenda of 21st century at that time. And this is actually a comprehensive plan of action to be taken globally, nationally, and locally by organization of the United Nations systems, governments, and major groups in every area in which human impacts on the environment. In other words, to say this very simple, so from first time in 1992, humans became aware that we have to do something in order to sustain us on a planet. Yeah. To start thinking how to balance humans and nature. This is my personal perspective, how I see that. I will be focusing only today on Convention on Biological Diversity. So biological diversity means the variability among living organisms from all sources, including terrestrial, marine, and other aquatic ecosystems and ecological complexes of which they are part. So that means we are talking about diversity within species, between species, and of ecosystems. The Convention on Biological Diversity is actually international legally binding agreement that has three major goals. The first is conservation of biological diversity. The second is sustainable use of its components. And the third that I will be talking today in detail is the fair and equitable sharing of the benefits arising out of the utilization of genetic resources. I would like to give you some definitions here. So when we are saying here genetic resource, we mean that we talk about genetic material of actual or potential value. Genetic material means any material of plant, animal, microbial, or other origin containing functional units of heredity. So we are not talking here about human material, just to be clear. Convention on Biological Diversity has so far 193 parties, and there are 3 protocols that happened so far during this history from 1992. First is Cartagena Protocol, second is Nagoya Kuala Lumpur Supplementary Protocol on Liability and Redress, and the third one is Nagoya Protocol on Access and Benefit Sharing. Cartagena Protocol deals with, it's again the international agreement, and it deals with living modified organisms. So the aim of this agreement is to protect biological diversity from the risk posed by genetically modified organisms. The second one, Nagoya Kuala Lumpur, came after this first one, and it's a supplementary to the first one. And to say it very simple, the aim of this agreement is to ensure the safe handling, transport, and use of genetically modified organisms. And it actually entered into the force in 2018. As you can see by years, so all these processes is actually very, very fresh. Yeah, they're happening now, although they started so many years ago, so many decades ago. The last one that I will be talking about, not the last, but that I'm gonna talk now, is Nagoya Protocol on Access and Benefit Sharing. Nagoya Protocol on Access to Genetic Resources and the Fair and Equitable Sharing of Benefits arising from their utilization to the Convention on Biological Diversity is an international agreement which aims is to share the benefits arising from the utilization of genetic resources in a fair and equitable way. And this agreement entered into the force in 12 October 2014. What I'm talking here about. So what is this story about access and benefit sharing? That means that each country on a planet that is a part of this agreement, and there, as I said, it's almost it's 190 something countries. So each country has sovereign right over the genetic resources that are found on their territory. When benefits arise from research or development of this genetic resource, including when it leads to the commercial use of a developed product, these benefits should be shared fairly and equitably with the country providing this resource. So we have this Convention on Biological Diversity that protects biodiversity, and we have this Nagoya Protocol that which aim is to help us to how to access and how to share the benefits of genetic resources. Let's make it now more clear. So this access and benefit sharing is actually, we are talking here about taking something and giving, giving and taking. Access means that user We have all in this situation, we have a user and we have a provider. Yeah? User seeks access to genetic resource, and he is, or she or institution is his aim to access the genetic resource is because of some scientific research, or user wants to develop a commercial product. What does it mean now, benefit sharing? It means that you have to, because you took something, you have to give something back. So provider of genetic resource grant access, and in exchange for a share of the benefits that results from their use, so they have to get something back. And at the end, this all should contribute to the conservation and sustainable use of biodiversity. Uses of the genetic resource may be represented by, as I already mentioned, by basic non-commercial research, which means you want just to determine, I don't know, taxonomy of your species. You have some super fancy plant that you have, or fungi that you found in a nature, and you want to do taxonomical research, and yeah, you start collaborating with somebody outside of your country, and then you have to think of this access and benefit sharing. Or you're doing a commercial research in, I don't know, biotechnological, pharmaceutical, whatever. What does it mean, these benefits again? Benefits are arising from the use of genetic resource, and benefits can be monetary or non-monetary. Monetary benefits are benefits that comes from the research and developments of some commercial product. That means that you are getting some royalties, milestone payments, licensing fees, and so on. Non-monetary means that you think of some technology transfer, enhancement of research skill, you make some joint scientific publications, and so on. So the final aim and goal of the success and benefit sharing is to contribute to the conservation and sustainable use of biodiversity of each country on the planet. And at the end, it can also contribute to the poverty elevation and sustainable development in general. So coming back to again to this definition of genetic resource. As I said already, genetic resources are material that can be of plant, animal, microbial, or any other origin, but not human, that has actual or potential value and contains genetic information. But here we are not speaking only about the DNA, we are also here speaking about derivatives, as well as traditional knowledge that are associated with genetic resources. Of course, there are also some material that are outside of scope of access and benefit sharing scope. This is a material that is covered by some special instruments. For instance, when we are speaking now about coronavirus, if you will, then different laboratory strains, pathogen or pests, or sequences that are obtained from public databases, if we have some synthetic molecules or derivatives that are not connected to the genetic resource and the original derivative. So when it comes to this legal part, what does it mean, the success and benefit sharing? It means that the user that is seeking for access to genetic resource must obtain so-called prior informed consent. So he has to get permission from the provider country. And this permit states what the applicant may collect, when and where. And this document is issued by the competent national authority. On the other side, the provider country and user country have to have an agreement about the benefit sharing, and this is called mutual agreed terms. So it defines how you may use actually your collected material. It is very important for you to know if once you are connected to this topic, that first place where you need to look and to go is this link that I have put here. This is a platform for exchange of information on access and benefit sharing for any country in the world. So you can really find there very, very valuable information and get, if you're like, want to come, I don't know, to, let's say, Germany, then you can find even the names and contact persons, yeah, for different departments in a country to get. Let me now summarize that through using this graph. Yeah, so we are having a genetic resource. So this genetic resource can be found in a nature, so in a natural habitat, or this can be a part of some botanical garden or university collection. Yeah, so you're at your university, you have some super fancy, I don't know what, for instance, fungi, and you have a so-called provider, yeah, somebody from other country wants to use your fungi, or he wants to use a knowledge that you have about that fungi. So then we're talking about this traditional knowledge. And you want to, yeah, you are a user, yeah, you have to contact your provider. So we are talking about here about providers and users. And you have to exchange these documents, so-called prior informed consent and mutually agreed terms. Once when you do that, you can start utilizing your genetic resource for commercial or non-commercial use. And at the end, when you will have a benefits, and these benefits can be monetary or non-monetary. And yeah, so this is at the end the overall picture of this access and benefit sharing. And how now, I will give you two examples how it looks in practice. So here is an example about fungi that was found in Benin, so in Africa. And Africa was provider country. And on the other side, Germany were very interested in determining this species. And they made a collaboration, scientific collaboration. So they transfer even material from Benin to Germany. They signed all of the document, and at the end, they had some joint publications. Next example is more interesting. It's about traditional knowledge of the, I have no clue how to pronounce this, Hoodia plant. This is a very juicy indigenous plant of southern Africa. And this plant was used by century by indigenous San people, yeah, southern Africa people, to stave off hunger and thirst. In 1996, there was some research and
Halo, sorry Sanya. Halo, Mas Beni. Okay. Sorry Sanya. Okay, you can continue.
May I continue? Yeah. In 1996, they made they patented some compounds from this plant. Yeah, they did commercialization, but without involving any Sun African people. Yeah, and it was like scandalous. And at the end, they kind of like refer to this Nagoya Protocol, access and benefit sharing, and they managed like to really to sign some agreement. And today they observed benefits, yeah. And they even got two types of benefits, these monetary benefits, which means milestone payment, as well as royalty payment, and also non-monetary, means different funds for development, education, and training of the San communities. So it was really like a very, very good example. I also did a little bit of research about, I was interested to find out what is the situation with your country. And I'm very well aware, and I think that everyone on a planet, that Indonesia is one of the mega diverse countries of the world, especially with these hotspots. Indonesia is a part of Nagoya Protocol, so you ratify on this date presented here. However, there is still so many unclear situation happening in your country, but it's not only in your country, it's everywhere. For instance, as a Germany, as a country with a very good economy, and everything is still start implementing this Nagoya since 2016. Yeah, so very recently they started. So it's really, I would say, very new for everyone, not only to the East or West countries. So everyone is kind of new to this procedures and protocols, and it's difficult even to understand them for authorities. What I have found about your country, I was not able to read everything because I couldn't translate in English, but what I found is that there are several regulations already existing. However, these regulations that are existing that are published by government, yeah, or very official documents or law, if you will, only applies if you on access. Yeah, so the currently government is regulating access rights, which means that, for instance, foreigner, yeah, some user, other country, can conduct or transfer the material, but any biodiversity genetic resource from your country to outside or traditional knowledge. He can do that only if he get the license from the national government. Yeah. And this is called the material transfer agreement. So this is not the document that I was mentioning before. What is missing right now is the existence of the establishment of benefit sharing agreement, which means that you really have to have something that regulates how you can get a benefit of the material that you gave granted access to. And I hope that there's still, I found some article which is very fresh, I think from last year, that is talking about giving some good examples. So that is really things are moving in Indonesia, but still it's very slow to get contact with some local people, how to say. Maybe there is already awareness of the high level education level, but not really in a praxis. And it's a long-term process, but it's good to start. And I invite all of you as educated people to think of that and to get in contact and to protect your biodiversity, and at the end to benefit out of it. I would be happy if I manage with my lecture to give you some impression. What does it mean, strain collection, microbial strain collection, how to build it, how to utilize that. And overall, microbial collection is important bioresource that supports development of life science and not only fishery and marine industry, but any bioindustry. Microorganisms at the end, they really contribute to the health of human animals, plants. They help solve us also food problems and help us solve environmental problems. On the other side, I would be also very happy if I managed to transfer you this important information that you have to, when you're working with a bioresource, that you have to think about your rights and think about contacting always national or local authorities and try to regulate access and benefit sharing. Here you can find my contact information. If you have any question, I'm really very helpful to answer them. Thank you. With this, I'm
Okay.
closing my lecture.
Okay, Sanya, thank you very much for really the great presentation today. Really nice lecture. Now we have like a short discussion session. Is it fine?
Yes, of course.
Okay. Okay. Okay, if anyone have any question, comments, or maybe also suggestion to Sanya, please raise your hand, or you can kindly write your question or comment to this chat box. Maybe someone want to ask directly. Please raise your hand. So here, it would be nice if you on your camera, and then we can have like interactive discussion today. Maybe for my student, if you have like sometime my student they feel not sure for their English, maybe you can ask in Bahasa Indonesia, then I will try to translate to English. Maybe from student or other audience, any question related to the Fraunhofer collection, how they manage the so many microorganism collection in Fraunhofer. I don't know how busy Sanya at the moment. Today we also have a streaming via YouTube. Maybe you can also write your question in YouTube also if you could not access the Zoom because we have limited access today. Okay, while waiting the question might be from student or from the other audience. Actually, at the semester we have course of marine microbiology. That hello, anyone want to ask directly? I heard someone open the mic. Okay, maybe later. Okay, Sanya. In this semester we have course of marine microbiology. That you know that you talk also that Indonesia one of the mega diversity of in the world. So of course we have really high diversity also of microorganism. In our university, now we are focused on marine bacteria. Yeah, maybe maybe you can explain more detail. Maybe for a student, you talk about the culture dependent dependent and independent. Maybe you can give more explanation.
Overview, yeah.
Yeah.
Okay, so what does it mean culture dependent, culture independent? Because very often, so we all are looking for, you don't want, when you do some isolation project, you don't want to isolate something that somebody isolated before you, yeah? You want to have something that is unique. that is, you know, coming for the first time. And for instance, you are interested to isolate something that so far was not isolated in terms of it was not culturable. And with this modern era, modern technologies, you can obtain by using this culture independent method, meaning you can by DNA amplification, actually you do 16S amplicon, very, very simple. You can get a very comprehensive picture of the whole microbial community. Yeah, you took, I don't know, invertebrate, and you want to find out composition of yeah, microbiome, gut microbiome, and then you isolate total DNA and do 16S amplicon sequencing, and you find out actually the complete diversity. You observe the complete picture of microorganisms that you have inside. And based on the proportion of microorganisms, then if you want to have that on a plate, on a really in your hand, how to say these microbes, then you have to come back to this culture dependent method. Yeah, but now you have a little bit some knowledge how to do that. Now you have some ideas. If something is really very hard to cultivate, which is very often the case, that these are the stuff that you're looking at, then it's very challenging. Yeah, you have to really think a lot, which type of media, what supplements to take, what not to take, and so on and so on. So very, very challenging process. But at the end, once when you succeed, then you can benefit a lot.
Okay, thank you, Sonia. Yes, that we also tried these two methods in our previous laboratory. Okay, hopefully student and all audience can get the point of that Sonia give explanation. Okay, I open again for the discussion session. Any question? Usually the student are active in the class. Salwa, hello. Usually you are active in the class and has the critical question. Hello, Salwa. Can you open your mic and turn on your camera? Hello. Hello. Maybe if you have question in Bahasa Indonesia, is it fine?
Yes, of course. For me, it's fine. You can they can ask on your language, and then you simply translate, and then that's fine.
Yeah, of course. Yes, anyone, if you have question in Bahasa Indonesia, you can also directly raise your hand, or you can also write in the chat box, or maybe ladies and gentlemen that are watching via YouTube, you also can ask write your question in the YouTube. Okay, maybe someone still find the question, or maybe also Sanya already gave the really clear presentation, then everyone can understand everything. But actually in the last presentation that you talk about the Nagoya Protocol, in the last year before I back to Indonesia, we have some, like, maybe you can also tell to us what happened with our sample related to the Nagoya Protocol. That I have the marine bacteria from the sponge that I will keep those samples to Fraunhofer building, to the Fraunhofer marine microorganism collection, but because of the Nagoya Protocol, it could not.
Yeah, because there was actually, I think what happened before even you started this research, that there was a contact, I think that Til Scheberle, he wrote to Indonesia local authority office, asking for a help, yeah, for understanding how to proceed with samples. Is it possible to do a research outside? But so far he never got any actually reply. Yeah, so still on a waiting. But I think that, yeah, he should try to fight a little bit more, and also maybe I can take over that, and a little bit with your help, maybe to write, because I think he wrote in English. Maybe it would help if you write like on a local, like your language, and maybe get some information from local authority first.
Okay. Yes, the point at the moment related to the Nagoya Protocol is for us, maybe the information from you. Sorry. I should close this message here. Some information in the chat box. Yeah, regarding to the Nagoya Protocol that you already presented today is really great information to us. Here's Sanya. Sorry, one question goes to my email in Bahasa Indonesia. Wait, I will try to translate it. Okay, so the question was, so if we have like 100 maybe or 1,000 collection, she said about the bacteria collection, would you please recommend with one of the genetic tool to screen maybe in the more efficient or fast, maybe related from your slide? You mentioned the RAPD or maybe
yes.
box R that you already did for me.
Yeah. Depending on depending on a purpose, yeah. Depending, you will use, if you want, like genetically to exclude redundancy.
Would you please share also your PowerPoint? Then we can also overview.
Just a second. I just have to find it.
Yes.
Just
yes.
So what is good about this method? You don't need to isolate DNA, yeah, because you're doing like amplification. Yeah, you need a DNA, but you don't need a pure DNA. Yeah, this is the first thing that is very like you are already on a in plus, how to say, already like you have some jump start. That means that only what you have, like you have your cultures, your microorganisms, and it's really enough if you only lyse the cells. Yeah, you can lyse the cells by some mechanical lysis, or you can use lysosomes, like mechanically, chemically, whatever you want. And this is like enough good enough material for doing this reaction, PCR reaction. And depending on a type of microorganisms that you would like to determine, so you already have some knowledge what is inside because you applied already specific media, yeah. So you are you have some already previous knowledge. And then if you have, for instance, actinobacteria, the best way would be to use box PCR. Yeah, if you have gram-positive bacteria with low GC content, then it would be good to use some GTG primer, so that amplifies these GTG regions, repetitive regions in a genome. Or if you want to work with some more like this interaction proteobacteria, enterobacteria, then you would use these primers that are very specific. These repetitive elements are very specific for this group. Or you would like to use so-called random amplified polymorphic DNA primers that randomly amplifies DNA, yeah, without any connection to anything. And by, yeah, so as I said, it's not so difficult because you already know in advance what you have in. Of course, you never know completely, but at least you have some pre-knowledge, you have some starting point, and if you see that this is not working for you, then you can always start with other primer set for the other group, yeah. And what is disadvantage of this? It only applies to bacteria, so you can't do that with fungi. Yeah, because here we are talking about prokaryotes. And we do that, you can do this very simply. Yeah, you have your cell lysed, you do PCR reaction, and then you can run very simple gel electrophoresis, or you can use some fancy equipment as I showed here with this microchip, very like miniaturized system. And if after like reaction you observe the gel, yeah, fingerprinting gel, in our case it's very well, it is like with the software digitalized, but you can observe normal gel, you can take a picture of the gel and then use any software to count intensity of this DNA bands that you observe. And then you look at these genetic profiles and you look at this PCR bands, and you try to find out how similar are they. If they are identical, in many cases that means that if they're identical, then they are the same. But there are very rare cases in nature where they are not the same, because you can have like always, yeah, this environmental factors that can invoke that, I don't know, one gene is expressed, I don't know, 10 times more in that strain than in the other one. There is no guarantee, but as I said, it's very low in nature that you have these examples. And yeah, most of the time you need you can do that manually, but it's very difficult if you have a huge amount of samples. Then you need some digitalization.
Okay, thank you, Sanya. So it's really advantage method because we don't need the pure DNA for this.
That's a real advantage, because you can do that really fast. Yeah, it's very, very convenient.
Okay, maybe for the all student and other RDN, maybe it's really the recommended method, because we usually have a problem like yeah, of course, for the DNA isolation kit, here is quite expensive in Indonesia. Yeah, then, but this is only specific for only bacteria, ya, Sanya, ya.
Yes, that's a disadvantage. This is disadvantage. I mean, there is so many different ways. If you want to look at the end, this is a DNA polymorphism. Yeah, so here we are speaking about DNA polymorphism, and there's very different ways how you can look at it. You can, but this is I would say the cheapest way. Yeah, the cheapest in terms of material and in terms of speed. You can also speed, maybe not. How you can still determine DNA polymorphism? You can you can apply to your DNA samples restriction enzymes. Yeah. And then you can do like this restriction digestion, restriction enzymatic analysis, and then again you observe the bands. Or you can do, yeah, this would be the most common methods, like restriction analysis or this amplification by DNA. No, nothing else is coming to my mind.
Yeah, but
they're
everything is clear,
different.
yeah.
There are different ways, you know, how you can you can do that. Yeah. So
Yeah.
when we come to talk about DNA polymorphism, it's really like a never-ending story.
Yeah, okay, but everything is clear. Hopefully, the student also understand. But later I will translate to my student that send message, private message to me. Okay, maybe I will give one more session if anyone want to ask directly, or is it fine if you want to send message, a private message to me also? Okay, I think question in the chat box hanya, but in Bahasa Indonesia. Let me read first, then I will to make summary. Okay, in briefly, Corey Hilmi asks, so that we know, Indonesia, we have like a limited funding, limited high technology for like molecular research. How to manage this kind? I don't know, is it this our topic or not? But maybe you have like another option, the cheaper molecular research. Then if we are working with the molecular step, how we can make sure to the people or the user if our product is safe.
I'm not quite sure that I understand. If you're working on a molecular level,
usually we have problem to make sure to the last user or the two people this our product is safe. Maybe
how do you mean? I didn't understand. You have a product.
Maybe from the, like, for example, genetic modified, yeah, from the GMO, then they will produce like the different product from the natural. How we can make sure to the people if our product is safe.
If you have something genetically modified, how you can make sure that that's safe. That brings me back to the presentation. Yeah, I talked here. Let me share again a screen. Yeah. So this is, at least, I'm sorry, I have no clue how it is outside of the Europe, but I know for the European Union, yeah, how it's regulated. So you have this second protocol, yeah, called Nagoya Kuala Lumpur, yeah, which deals with these living modified organisms, genetically modified organisms. And then you have like each country in Europe has developed like a set of rules how you can deal with genetically modified organisms. And at least in, now I'm talking about Germany, you have to have, like myself, I have a training as I had to do yeah, kind of like to educate myself through different seminars and do some schooling, how to become a genetic safety project leader. And it means that if you want to do a research with, to do any genetic modification research, you have, each step of your work has to be controlled. Yeah, so you have like really to document each step you do in a lab. So you write usually in your lab journal what you did with your experiments, but in our case, you also have to write that for the official authorities. And once a year, we have a control that is coming to our institute and looking to our documentation. And now they said, no, this is not enough for us. From next year on, they will simply come to our lab. They will take physically a sample from us, bring to their laboratory, and amplify, because okay, of course we will say with which primers and what and how, and then they will control, do we really have what we say to them that we have? Then they now do not believe us with just giving them documentation. They want to go deep and to find out themselves, is it the thing that we are having really what we are is supposed to be or not? So it's very, very complex system. And I think it doesn't exist in all countries. For instance, I know for the United States, they don't, because they are like selling like crazy this genetically modified food. But you can easily always check that by amplifying a certain thing and comparing with like original sample. I hope I answered the question if it was going in that direction.
I can understand. Maybe I will translate later. Maybe this is the first question is really common for students. Maybe what are also researchers in Indonesia, that if we want to work on the molecular level, usually we have problem with funding and the equipment. Which one that is the really cheaper that you recommend to us? It's really hard to answer.
Yeah.
Because
Yeah.
everything is in Germany is really, we can just take in the lab.
Yeah, but this is, I would, I mean, you as, it doesn't mean, I really like me personally, I appreciate the facilities who doesn't have so good like financial situation, because then you are very creative. Yeah. So you really have to implement, you really have to put a lot of effort in order to observe some results. Yeah, maybe you take some very long steps, but at the end you can, you still can do the things that I was talking to do this characterization without this genetic stuff. Yeah, you can skip this molecular biology. Come on, the majority of our collection is coming from era where molecular biology tool was not present. Yeah, so we have, as I said, we have samples from 1940. Yeah, in 1952, 1964, and so on. People didn't have PCR. Yeah, PCR appeared in the 1990s. Yeah, let's say so. I think it was 1990s. So the period before, there was no any fanciness or something. People were just having like normal light microscopy, doing stuff. And what I can tell you, the work they did is, I don't know how many classes better than the things we are doing now. I don't want to offend anyone of us, but I really do appreciate a lot of work from people, how people were doing things in past. And if you will, in past, I mean, now it's a little bit more philosophical question. In past, we didn't have these digital tools, and we simply had more time to think. Yeah, today you are working or you're studying, and then you go home or you go out with your friends, and you're checking your emails, you're checking this and that. You don't have a freedom to think. You're constantly occupied by some digital information and work around. You simply don't have time for yourself without a computer looking at some screens and really think of the things you're doing. So maybe during the studies are different, but if you are not studying, that's the truth, I can tell you.
Okay, Sanya, we also get the other option of that Sanya give to us to give motivation to us. Yeah, we can also doing this research. Yeah, this maybe sometime yeah, we can be creative for our research. I believe the all student, all audience here can do that.
But this is what is also important to make collaborations. Yeah, that's it won't hard. It will be helpful. Yeah, because as I said, you are the most mega diverse country. If it's not on a first place, on a second, I'm not sure, but I would put you on a first place. You have so many things to offer. Yeah, you have sources, but you don't have financial support to all that, and then you can search that outside of the country, and and everyone can benefit. Yeah, the community in general, with the new knowledge, new products, and you as well.
Yes,
Just
yeah,
I would
that's
really encourage all of you to try to make collaboration. There is so many things ongoing. Yeah, so many different fund agency that are supporting collaborations, international collaborations.
okay, Sanya. That we are waiting for the word collaboration. That also the first of opening remarks, our dean, SD, also mentioned that maybe in the next after guest lecture is the collaboration with the Fraunhofer Institute in Germany. Okay, all of student and all audience, maybe finally we come to the end of our lecture. I will close this session. So maybe I can give short summary for our lecture today from Sanya, that we know about the Fraunhofer collection composition and usage of different bioresources for isolation and from microorganism. Sanya also gave introduction biobanking in general, strain management and importance of quality criteria, and also touching a lot of Nagoya. That is really important for protection of its country diversity. Okay, in the end, I would like to say thanks to the guest lecturer today, Sanya, for the interesting presentation, and also for all my students, all the audience, for the participant today. Thank you also for the two of my students that already asked for some question today. It's no problem. Yeah, today we also learn about not only marine microbiology, but we also can practice our English. Hopefully this lecture today will be beneficial for everyone. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh. Okay, Sanya, thank you very much. Yeah, see you maybe.
See you.
Yeah.
Bye.
Bye-bye. Next time, anytime, anywhere. Yeah, thank you for all. Okay, we can close this public lecture today. Thank you very much for all. See you for the next guest lecture. Yeah. Yeah, from student. Thank you. Thank you, Dr. Sanya. Thank you.